Hey everybody, how you doing? It's Tuesday afternoon. I'm filming, but this will be posted Wednesday morning, March 25th. Before we get going, I want to go over the chapter 23 reading guide for today's lecture, if you will. But before we get going, I have a couple of announcements. First is my verbal joke of the day. So we'll start there. If a child refuses to go to sleep during nap time, are they guilty of resisting arrest? I'll let you think about that later. Let's move on. So, announcements for you guys. Next Monday, we're looking at a chapter 22 quiz. And next Wednesday, we're looking at a chapter 23 quiz. So, we're basically wrapping up our introduction to macro and microevolution. And so, question came up about the format of quizzes or tests. That's still in the works. I'm not positive that I'm going through Schoology as I'm trying a few avenues to test you guys best. But you are going to see some review problems that you can work on as you prep for the quizzes next week. Format will be generally the same as what you've seen in the past from me, a mix of multiple choice and short answer type questions. Keep in mind, there probably will be some calculations, especially when it comes to Hardy-Weinberg. So getting your head wrapped around those concepts and those calculations is important. You will have one more Hardy-Weinberg lab type activity to do on this Wednesday, March 25th posting. Otherwise, for today's submissions, for Wednesday's submissions due Thursday, you're going to have an activity on Hardy-Weinberg equilibrium, a lab of sorts, like I said. And then you'll also have a short worksheet to work on in terms of understanding the differences between the different types of selection and the changes that can happen in a population based on environmental pressures. So I think that's all my major announcements that I wanted to get going on. Let's hop right into chapter 23 reading guide. As I left you guys off the other day with just 1 through 13. So here is starting with number 14. I'm just going to kind of cruise through these, and then I do want to get this posted for you guys also on this Wednesday. So there may be a variety of things posted in that folder. Try not to be overwhelmed. I'm not trying to overwork that part of you guys, but there's going to be some good resources for you there. Number 14, we have to nail down the definition of what a population is. This is a group of individuals of the same species that live in the same area and interbreed, producing fertile offspring. So they are organisms of that same species that are able to produce fertile offspring. Then as we work our way into this Hardy-Weinberg understanding, a population's genetic makeup is its gene pool, which consists of all copies of every type of allele at every locus in all members of the population. That's an insane amount of information. So what we do as we really break down this understanding of a gene pool is we spend time looking at just like one gene, as you have noticed as you've worked on these Hardy-Weinberg problems. We know that if there's 2 or more alleles for a particular locus in a population, we know that individuals can be homozygous, meaning that the 2 alleles are the same, or they can be heterozygous and the 2 alleles are different. As we go down, I'm not going to walk through this example as you've had a chance to work through some problems already, and this really is pulled straight from your textbook and was a fill in the blank to at least process some of the calculations that are done. The math is not difficult. It's I think what gets kids tripped up or gets you guys tripped up from time to time is what step should I take based on the information that I have in front of me, because the information isn't always the same. So there isn't just a lockstep order of steps that you have to take every single time to get your answer, quote unquote. Things that we do want to pull from these questions here are that we use the letter P always to talk about the dominant allele and the letter Q for the recessive allele. It doesn't really make that very clear here in this example because it isn't always a matter of straight dominance and recessiveness, as this one up here was an example of incomplete dominance. So we just delineated and made sure to call one allele its frequency P, and the other allele was frequency Q. So whenever we're talking about P's and Q's, it is we're talking about the frequency of alleles or the frequency of genotypes as we get into the other formula that we use. So as we go down, what we have to understand, and we'll get into that in these next couple of questions, is we have to get at least wrapped around our brains a little bit what Hardy-Weinberg equilibrium is for and what we use it in connection to. And it's basically this null hypothesis of sorts. that, as we see in 21, it says, what is one way to assess whether natural selection or other factors are causing evolution? What we have to do is determine what would a population look like if it were not evolving, if no change was happening. And so that way we can quickly see, even with just a little bit of something factoring in, like selection or migration of some individuals moving in or moving out of a population, how that will change allele frequencies. And that change in allele frequencies will mean that evolution is taking place. So Hardy-Weinberg equilibrium, as it says here, in a population that is not evolving. What I didn't say in there is that this is a pretty hypothetical situation. It is rare in nature that you're going to find a population that really isn't changing its genotypes or its allele frequencies from generation to generation. The only reason that really happens is if the environment is stable and the alleles are pretty fixed, meaning there's really only one allele that's running that genotype. So it's a population that's not evolving. Allele and genotype frequencies will remain constant from generation to generation, providing that only Mendelian segregation of alleles and recombination of those alleles are at work. That population is in Hardy-Weinberg equilibrium. Let's see. I didn't fill in the diagram in 23, as again, that was just pulling information from the internet, from your textbook. But the question says, if the population is in Hardy-Weinberg equilibrium, what will happen to these frequencies from one generation to the next? Again, the allele and genotype frequencies will stay the same from generation to generation, because these things that cause change in this hypothetical population, we're saying that they're not occurring. This is this idea of this control population to compare actual changing populations to. As we look down at our other formula, our p squared plus 2pq plus q squared, we just are defining terms here, as I mentioned as I went over practice problems 1 and 2 the other day. P squared, that is talking about the frequency of the homozygous dominant population. As we move on down, 2pq is the frequency of the heterozygous population. And finally, q squared is the frequency of the homozygous recessive population. Things to clarify. When we talk about frequency of any of those, the heterozygotes or the homozygous dominants, etc., frequency of one of those is going to be a decimal, like 0.495. That is just the decimal before we say change it to a percentage. So whenever you're asked for a frequency, if you're going to answer it according to the letter of the law, you're going to give it as a decimal answer. These conditions that we talk about to keep a Hardy-Weinberg equilibrium, again, in this hypothetical population. So we can see right away as we look at this list, we're like, that's weird. Like, where would that ever happen? Exactly. That's the point. It's not that we say that this happens. It's something to compare to for those populations that do change. We note all of these things that can cause change. So for instance, in a Hardy-Weinberg population that's not going through evolutionary change, there is no mutation. There is only random mating, meaning whatever male a female bumps into, boom, they're having babies. There isn't choice of mate so much. Number 3, there is no natural selection, meaning there is no advance or increased fitness or ability to survive of one individual over another in a population. They all have the exact same fitness level and ability to survive. Number 4 covers this term that's come up a couple times, which is genetic drift. This is an extremely large population, so this keeps from major fluctuations in frequencies that can happen because of genetic drift. So if a hurricane comes through and hits a population of 4 billion beetles and it kills 100,000 of them, that's still a pretty big number, but it may not be enough to cause major changes in allele frequencies. So that's that extremely large population. Finally, no gene flow, meaning that there is no individuals moving into or out of this population that could potentially affect or change allele frequencies. Number 26, the answer was right in the book. But Q is just reminding us that is the frequency of the recessive allele, and Q squared then is the frequency of that homozygous recessive population, or 2 recessive alleles. Q squared was 0.0001, and all you needed to do is take the square root of that number to get Q. So in that population, the frequency of that recessive allele that causes PKU was 0.01, or just 1% of the alleles in that population. Again, quick pause there. As you're going through these numbers, again, It's not so much the math that's going to trip you up. It's knowing how to approach a problem and see what information you have and what are you trying to answer from the questions that are being asked of you. Finally, what I tell you guys regularly in class or here on the internet is the frequencies that you're seeing, those are just percentages. So when I see that Q equals 0.01, 1% of the alleles in that population are the recessive allele. That's what that mathematical equation is saying to you. As we go down, we move into 23.3, which is natural selection, genetic drift, gene flow, all these things that can change allele frequencies and violate Hardy-Weinberg, which can cause evolution. So DDT was a pesticide that was used many decades ago, and it worked really well early on. Killed off pests, crops thrived, but over time, DDT worked less and less and less. Basically, there were pests that were surviving DDT. They had resistance to DDT. And they were the ones that made babies, and they passed on that resistance to DDT, which is a similar example to talking about bacteria resistance or antibiotic resistance that bacteria have back in chapter 22. 28, in regard to coin flipping, the smaller the number of coin flips, the more likely it is that chance alone will cause a deviation from the predicted result. My quick example there is if I flip a coin 10 times, you can absolutely often see 5 heads, 5 tails being flipped in those 10 flips, but you also very often can see 9 heads and 1 tail, or 7 tails and 3 heads. Because there's only 10 flips, and that's a small grouping, a small sample size, we've deviated from the predicted result pretty largely. But if we flip that same coin 1,000 times and we're expecting 500 heads and 500 tails, we're going to get much closer to that expected result. It would be rare to see 400 heads and 600 tails, which is off by a bunch. We're not going to see that much deviation as we get to larger and larger and larger sample sizes. 29, that definition, chance events that cause allele frequencies to fluctuate unpredictably in a small population is called genetic drift. The example of if there's 10 beetles sitting on the floor and 3 of them are green and 7 of them are brown, and somebody walks by and accidentally steps on 3 of the beetles, that's going to change the allele frequencies real fast. And no beetles really had an advantage of surviving over another. It was just through a chance circumstance that caused some of them to get stepped on and be removed from the population. The 2 examples in 30, founder effect and bottleneck effect, are examples of genetic drift. The founder effect is that if a few individuals become isolated from a larger population, their gene pool may not absolutely reflect the gene pool of the large source population. So therefore, those allele frequencies in that smaller population are different, and that's genetic drift. That's a changing of the allele frequencies just to a chance. Bottleneck effect is similar, but it's just the example of a severe change in the environment. And that will cause a drastic reduction in the population. Whatever survives is by chance, not due to selection, not due to some individuals having increased fitness or ability to survive. Just some survive by chance and some don't. That's effectively probably going to change the allele frequencies of that population too. 31, gene flow, that is just migration, movement of alleles into or out of. So if you hear the word immigration with the letter I, that's individuals moving into a population, whereas emigration with the letter E, that's individuals moving out. I'm going to skip 32. Just another example of movement of individuals and how that can affect allele frequencies. You can take a look at it for just another example to help wrap your brain around, but I'm going to move forward into 23.4 and talking a little bit about natural selection and how that can change populations and the genetics of populations over time. So relative fitness is this, and I keep saying that word fitness. Fitness is the ability of an organism to survive, thrive, and reproduce. So it's the contribution of an individual and how it contributes to the gene pool of the next generation relative to the contributions of other individuals. So if an individual has a high relative fitness, it means it has a better ability to survive and pass its genes on to the next generation than other individuals in that same population do. As I go down to 34, just these little graphs, I think have always, when I've learned this forever ago, they've stuck with me fairly well in that we're looking at changes that can happen over time. And we think about those bell curves that we looked at and sort of created through our first variation lab, where we noted that the extreme phenotypes, so if we were looking at, again, like bicep circumference or the size of your foot, we had, as you look at our graph over here, we had extremes, say very small foot size down here on the left and very large foot size up here on the right. If our y-axis was the number of individuals, then we didn't have as many at the extremes. And as we get towards the middle, you have a much higher prevalence. Many more individuals have that in between. So for us in foot size, it was that kind of that average foot size. As we look at this and we're looking at, it looks like mouse fur color. The lighter mouse fur color is one extreme and the very dark mouse fur color is the other extreme. Most of the mice have that intermediate fur color. So if we think about that as our starting place, the arrows that go down are pointing to potential changes that could happen to a population based on different environmental pressures and what individuals will survive best if things change. Because what we want to keep in mind with variation in a population is that allows or helps a population to survive best when the environment changes. So what we want to look at down here are three examples that we want to know are directional selection, disruptive selection, and stabilizing selection. Directional selection, I want you to think of the black line, the black curve, as present day, like right now, today. That's exactly what we have up here at the top, that black line. But let's say that something changes in the environment that starts causing the darker colored mice to just survive better. They survive better, probably mate with other dark colored mice that are also surviving better. There's just increased odds of that. So they're going to produce more dark colored baby mice and so on and so forth. So what we will start to see is where there used to not be a lot of dark colored mice, we start to see more of them being produced. We see more individuals that are darker. And what we see is over time, the red line is just looking down the future, 10 generations, 15 generations, just somewhere down the line, the curve will actually shift in a direction. It shifts in the direction where the extreme phenotype is favored, where one extreme phenotype is favored. So we call that directional selection. The next one is disruptive selection, where again, the black curve is current day right now, just like we had up above. But let's say an environmental condition comes up where the intermediate form, in this case, kind of the medium colored mice, they survive less well than the extremes do. So the darker mice and the lighter mice may survive better. So over time, we start to see a lot more of the lighter colored mice and a lot more of the darker colored mice, but less of the intermediate colored mice in the middle. So it's called disruptive selection. And it always made sense to me because the normal little bell curve that we get is totally disrupted. Now we get kind of two curves in there instead. So that's disruptive selection. Finally, same situation, but in this case, the intermediate phenotype, the medium colored mouse, actually in this environmental case, it survives best. It survives much better than the extreme phenotypes of the really light or the really dark colored mouse. And so we start to see more and more and more of that intermediate color. And so what we get is just a skinnying up of that curve, and it gets taller. We just get more individuals with that intermediate phenotype. And so down here is a brief description of each of those types of selection. Finally, the last couple. 35 is just talking about selection that can occur in a population when it comes to sexual selection. Sexual selection is really just coming down to choice of mate. If we're talking about actual natural selection occurring, there's generally not random selection, but non-random selection, where there is choice of mate based on whatever characteristics that population chooses. So the terms that I want us to know here are intrasexual versus intersexual. So intrasexual is selection within the same sex. So the example, or not example so much, but it's stated in the book that this usually occurs among males. And I wanted to read this piece to you in my 11th edition. This is page 497. This, again, is intrasexual selection. It means selection within the same sex, where individuals of one sex compete directly for mates of the opposite sex. In many species, intrasexual selection occurs among males. For example, a single male may patrol a group of females and prevent other males from mating with them. The patrolling male may defend his status by defeating smaller, weaker, or less fierce males in combat. I feel like that's talking about high school males, but it's really not. It's just talking about nature. It's talking about any form of nature that these traits are exhibited. A form of bullying is done, but it's done from a position of keeping that individual's genetic line going on and passing genes on to create offspring. And so the other one is intersexual, where we're speaking of mate choice, where individuals of one sex, and this is taken from the text again, it says usually females, are choosy in selecting their mates. This is not always true. I don't want to overstate the usually females comment, because this happens in different species where the males are very choosy in selecting females. And so that's where you'll see like a picture of a peacock showing off all of its colors and showing off how incredible it is, in hopes that a female will choose him for a mate. So that's intersexual selection. Heterozygote advantage. The example that's given is of sickle cell anemia. Sickle cell anemia is a lethal disease. It's a recessive disorder. So if you have two recessive alleles for sickle cell, you have a very tough life ahead of you. If you're a heterozygote, meaning you have one dominant normal allele, you're fine, but you display some traits of sickle cell. So it's not like a totally, completely okay, normal thing when you're a heterozygote. So what comes down, though, is that heterozygotes actually survive better when it comes to malaria, which is an interesting connection. But malaria, a totally different disease that people can get, people that are heterozygotes actually have an increased resistance to malaria. And so that actually helps people to survive in areas where malaria is prevalent if they have that one usually not good allele, that recessive allele for sickle cell, that's not helpful there, but it helps individuals to survive for malaria. So that's a heterozygote advantage. And then there's a list given at the very end of chapter 23, reasons why natural selection is not in the mode of fashioning perfect organisms. Humans are not the perfect organism. They're what we developed into with what raw material was there in the organisms in the ancestral species before us. And so, as it says down in the third bullet point that I have here, adaptations are often compromises. It only builds on structures that are currently present. It can't start from scratch. And so that's where it can't just be a population saying, man, it would be great if we had wings so we could just fly away from here. It'd be cool if we could evolve that. You can't evolve that way. Evolution is just built on the raw material that's there in the previous generations. And so those are the reasons why natural selection just keeps on churning, and it keeps on bringing about change that isn't necessarily predictable as things change, as we're seeing currently happening in our world today with COVID. The changes that are going to occur, even from a species and population perspective, scientists are probably working on predicting those things right now, but won't always really be successful with the changes that are going to happen to not just human populations, but to global populations of all kinds of different forms of life. So those are the main things that I'm really looking to hit with you guys as we wrap up chapter 23. Again, look through everything that's posted for this Wednesday, March 25th post, and please post questions on the page. I can't encourage you guys enough, since we're separated this much. This material is not always easy to click when we just try to learn it one-on-one. And as much as I want to give my time, and I'm giving it as much as I can to those that are asking for it, I also know that I'm not capable of sitting and speaking with you guys one-on-one and getting to each of you. So by posting questions on the discussion threads each day, that allows other people in the class, you guys, to take looks at that thread and help where you can. I've already put some points in the book for a few people that have stepped in and helped classmates with issues and questions that they may have. I'm leaning on you guys and hoping that you guys use that tool, because it will not only help you maybe with a couple of points, but it'll help you to understand the material better if you have to sit and explain it to a classmate. So thanks so much, guys, for working with me and just continually getting used to the system that we have here. You guys are doing awesome. Thanks so much. Miss you guys a ton, and I'll be in touch as soon as I can. Love y'all. Mm-hmm.